So we sort of mentioned the process of the cell cycle, but what we really want to get into next is the actual structure of the eukaryotic chromosome. All genetic material is called chromatin inside of the nucleus. But chromatin can take on two different forms. In its loosely coiled form, it's known as euchromatin. This chromatin is currently active, meaning that it has been that the genes are actually being transcribed at the moment. Heterochromatin, on the other hand, is going to be different. Heterochromatin is going to be inactive, meaning that it is tight and packed away and not currently being transcribed. And I want you to consider why this is. Euchromatin is really only going to be found during the stage of interphase. Interphase is when we have euchromatin. It's going to be loosely coiled and active. We're currently transcribing genes. We're currently synthesizing new DNA. At this stage, you won't actually be able to see the DNA underneath the microscope. Heterochromatin, on the other hand, is inactive chromatin. It's really densely coiled. I seem to be missing that bullet point, but make sure you put in your notes that's very densely coiled and inactive chromatin, meaning that the genes are not really being transcribed at this point. Really, the only time we go into heterochromatin form is for the sake of mitosis. We want to be able to move our chromosomes around through this process of mitosis, and that's a lot easier to do with tightly condensed inactive chromatin than the loosey-goosey euchromatin that we have everywhere. So we've covered the cell cycle as a whole, but we're going to cover each of the parts individually. Like we said before, chromosomes are highly regulated and organized structures. Cell biologists and geneticists have been able to construct detailed models of how chromosomes are organized. A eukaryotic chromosome contains a single DNA molecule, but it is actually at least 50% protein material. Some of these proteins are involved in making DNA, but most of them play a very important role, a protective role, by providing shape and structure to keep the DNA safe. These are what are known as histones. Histones are going to be proteins that DNA is going to be wound around. They're absolutely essential for maintaining our DNA, keeping it safe. The human DNA is roughly 2 meters long. That's really, really long. That's like 6 feet of DNA packed inside of one tiny little nucleus. The only way we are able to really pack it in tight is to wind it around these histones that can keep it wound nice and tight, safe and secure, and prevent any sort of damage from happening to it or getting it all tangled up inside of the nucleus. So the DNA is going to be wound around those histones, that 8 histone core. It kind of looks like beads. A string of DNA wound around DNA wound around histones is going to look a lot like beads on a string. Those histones are held together by linker DNA. That string is then going to fold into a compact zigzag structure and then form radial loops. At this point, it is still considered loosely coiled. Euchromatin represents the active chromosome that can still be transcribed by RNA polymerase and transcription factor. So this is what it looks like. So I know that was a lot of words before, but here's visually what that looks like. So the DNA this is going to be our double-strand DNA. It's going to be wound around those histone proteins. Those histone proteins are going to zigzag back and forth. Those zigzags are going to form these giant radial loops. At this point, this is what your DNA looks like inside of your nucleus during interphase. It's still kind of all loosey-goosey. It's safe, wound around those histone cores, but it still is kind of these giant loose radial loops. During the time of mitosis, we're going to condense those loops down into these tight heterochromatin structures that are going to make the DNA much easier and safer to manipulate and maneuver for mitosis and cytokinesis. So that's sort of an overview of the human chromatin, of eukaryotic chromatin. Next up, we're going to talk through the process of mitosis and cytokinesis.